WorldCommunityGrid.org members from around the world have been donating their spare computer time to accelerate research that helps humanity and the world. We thank all of our members for contributing over 2/3 years of computer time to the important projects on WorldCommunityGrid.org. About 7 years ago, the second research project to run on World Community Grid was FightAIDS at Home. This project is searching for drug candidates that can inhibit the HIV virus that causes AIDS. Please let me introduce Dr. Alex Pearman at the Scripps Institute in La Jolla, California. He will tell you about the latest science and progress in the FightAIDS at Home project on WorldCommunityGrid.org.
Thank you, Victors. I'm a member of Professor Art Olson's lab at the Scripps Research Institute. Today I'm going to tell you about the exciting progress we've made in the last year on advancing the discovery of allosteric inhibitors of HIV protease. Over 34 million people have an HIV infection, about 2 million people die each year, and throughout the entire epidemic, over 32 million people have died of HIV-related causes. That makes HIV the deadliest virus on Earth. Okay, today I'll tell you about how World Community Grid helps us all. Then I'll tell you about some background information, the exciting progress we've made, and then I'll tell you what happens after these calculations are done. So World Community Grid provides free resources from over 2 million computers for projects that benefit humanity. It advances research that seeks faster ways to cure diseases, find renewable energy materials, create cleaner water techniques, or develop healthier food staples. In total, over 165,000 CPU years of calculations have been donated to FightAIDS at Home. Also, over 16,000 CPU years have been donated to our Go Fight Against Malaria project. On FightAIDS at Home, we do virtual screens, which involves docking large numbers of compounds to try to find new inhibitors. So this is like trying to find master keys to unlock the control panels that can stop each piece of the viral machinery. But the keys and the locks are both flexible, they both change shapes as they wiggle and jiggle around, and they also have charged and polar parts that have to match up well too. To make it more difficult, there are often multiple different keyholes per lock, but they don't all work for the pharmacological effect you're trying to get. And over 10 to the 60th power different potential keys or chemical compounds exist in chemical space. Now, on these virtual screens, we can only screen thousands to a few million compounds, which makes it like not just searching for a needle in a haystack, but searching for a needle in a whole field of haystacks, and you can only explore a couple of those different haystacks. One of the main targets on FightAIDS at Home is Hiv Protease. Now, this viral enzyme cuts the viral multi-protein chain into separate pieces, which allows those pieces to fold up into their normal 3D structures, which allows the viral particles to form, mature, and infect other cells. If you sufficiently block Hiv protease activity, then that causes the infected cell to produce immature, non-infectious particles that can be removed from the infected patient. Most importantly, the introduction of protease inhibitors to the anti-AIDS cocktail significantly reduced the death rate associated with HIV. Now, this slide shows data from the New England Journal of Medicine, a slide prepared by Alex Vlodauer, and it shows in the red line that as more people started taking Hiv protease inhibitors in the mid-90s, then the death rate in green associated with HIV drastically decreased. Now, although there are many good drugs against HIV, infectious diseases, including HIV, almost always eventually evolve resistance against the drugs that are used to fight them. It's the nature of nature. To make it more complicated, HIV copies itself in a very sloppy manner. So many changes or mutations occur each and every day within each infected person. Now, if these accidental changes happen to help the virus resist the effects of the drugs, or if the lock's guts change and block the keys, then those mutants will have a selective advantage and it will spread. Now, since these mutant superbugs keep evolving and spreading, we need to discover new and better drugs to fight AIDS. Now, in the last year on FightAIDS at Home, we've been focusing on trying to find allosteric inhibitors of Hiv protease. Now, allosteric compounds bind in one site, and that regulates the activity of a completely different site. It's like putting a latch on the handles of a pair of scissors to prevent the blades from being able to open and close. Now, if we develop good allosteric inhibitors, that might help rescue the potency of current protease drugs against some of these superbugs. And in new combination therapies, it could have the potential to impede the evolution of new mutants. In Experiment 30 on FightAIDS at Home, we targeted two different potential allosteric sites, the 1F1 site on the top of the flaps and the 49 site on the sides. Now, the active site that we're trying to regulate by binding to these two allosteric sites is in this, the hollow tunnel in the center of the enzyme, shown there by TL3 in white sticks. The discovery of these targets were disclosed in that Chemical Biology and Drug Design paper from 2010 cited below. And we docked over 360,000 compounds against each of these sites and then fished through the results to try to find interesting compounds. In the first half of Experiment 30, we targeted the potential allosteric site on the top of the flaps, the 1F1 site. The top hits we found are shown as yellow spheres to indicate the general location of that binding site on the HIV protease. Now the active site is in that hollow center. So hoping to find compounds that can bind in that top site or on the side site to then regulate and shut down the activity in the center. Now we're showing these compounds as spheres or sticks with the same color because we can't yet reveal the exact nature and identity of these compounds until we've published the results, which we hope to do soon. Here I'm showing you 3 of the top hits we discovered against this 1F1 site. Now below it shows different types of experimental assays that our collaborators performed in order to figure out what these compounds do and how they do it. The DSF shifts, or differential scanning fluorimetry, indicate the amount that the compound is able to shift the thermal stability of HIV protease in solution. So 2 of these compounds displayed significant thermal shifts in stability, which shows that these compounds are able to bind to HIV protease in solution. Now BSI is backscattering interferometry, which indicates how the compounds change the overall conformational preferences of the target. Now 2 of these compounds displayed low micromolar affinity, but the other one didn't display a signal in BSI. So different compounds perform well in different types of assays, so that's why our collaborators need to do multiple different types of experiments to give us a good picture of what these compounds do and if they're really able to affect HIV protease. Now here I'm showing you the other 3 hits we discovered against this 1F1 site on the top. All 3 of these compounds displayed a significant thermal shift in HIV protease in solution. One of them displayed low micromolar affinity as well, but the middle compound displayed millimolar affinity, so 1,000 times weaker than the other hits that we've discussed. So obviously that middle compound isn't going to be as prominent in our future directions. Now so far against this 1F1 site on the top of the flaps, we ordered 10 different compounds, and 6 of those 10 compounds did display measurable effects on HIV protease stability and or displayed affinity in BSI or surface plasmid resonance studies. So a 6 out of 10 hit rate is rather good from a virtual screen. In the other half of experiment 30, we targeted the potential allosteric site on the sides of HIV protease. Here we're showing you the top hits as cyan spheres superimposed on each other to give you a general idea of where this site is. Now this 4D9 exosite is the allosteric site that I predicted might exist in the results of molecular dynamics simulations that I performed as a graduate student in Professor Andy McCammon's lab at UCSD and the Howard Hughes Medical Institute. And that was published in Protein Science in 2004. Here I'm showing you 2 of the top hits that we found against this 4D9 site. They're shown as cyan spheres. Now both of these compounds displayed a significant shift in the thermal stability of protease in the DSF assays. They also displayed low micromolar affinity in BSI, which is rather good since these are fragment-sized molecules and most fragments just have millimolar or 1,000 times weaker binding. So these actually are rather strong for being fragments, but it's still only low micromolar. Now very importantly, these compounds both actually inhibited HIV protease activity. At 100 micromolars, they only inhibited 9% or 6%, which means they're very weak inhibitors, but importantly, these are the very first compounds we've ever found in virtual screens against either of these potential allosteric sites that actually did display inhibition of protease activity in experimental assays. Here I'm showing you the other 2 top hits we found, these apex compounds versus that 49 site. These also both displayed a significant shift in thermal stability. Although they didn't, we were not able to detect any affinity for them in the backscattering interferometry experiments, these compounds both did inhibit HIV protease activity as well, although they're weaker than the first 2 compounds, which is why we're going to focus more on the structures of the first 2 compounds when we're trying to find larger, more potent compounds against this site. So far against that 49 site, we actually ordered 34 compounds, and 4 of those 34 did display significant activity in terms of shifting the thermal stability or changing the conformational preferences and displaying real affinity against HIV protease. Now, as we're searching for larger, more potent hits, one of the tools we can use to guide this process of finding larger compounds are the results of autoligand calculations. Autoligand is a tool from the Olson lab that's able to detect and characterize the features of ligand binding sites. In the yellow oval, you see the region where those 4 apex hits were predicted to bind, and so these autoligand calculations indicate we might want to find compounds that are extended down to the right or up and to the right. Now, the results of the previous dynamic simulations indicate that having compounds that go up and to the right should probably give us a better effect at being able to regulate the dynamics of this site to control the activity of the active site. So what I did is I used the Zinc server and I searched through to find compounds that had similar structures to those 4 apex hits, and then I docked those in calculations on the computers at Scripps. About 2,600 compounds I docked, and these 44 candidates I'm showing you here in cyan were the ones that performed really well in the docking calculations, and I recently ordered these, and our collaborators will soon test them. So as you can see, many of these compounds do have more of those atoms in the upper right direction, to allow us to explore that hypothesis about where the compounds should extend into in order to give us more potent inhibition. Now, the previous slides all showed a surface representation of protease. Here I'm showing you the space-filling representation, those white spheres. And this is to show that in addition to that upper right direction, there's also a potential trough we could try to explore in the upper left direction. So a few of those 44 candidates that I ordered do place a few atoms in that upper left direction to allow us to explore whether we might want to have compounds that go that way, or up and to the right, or perhaps in both directions, in order to give us much more potent inhibitors. So after, all this research is done as part of a large program project grant that Professor Olson's been leading, and it recently turned into a new HIV center, which involves multiple different labs in many different institutions around the country. So specifically, right now in the Olson lab, we're searching for new hits and designing new derivatives. The medicinal and synthetic chemistry labs of MG, Finn, and Valerie Fulkin are synthesizing derivatives of these compounds to characterize the chemical features that govern binding to these sites and hopefully inhibition. Bruce Torbett's lab and John Elder's lab are performing new assays to further characterize these hits, as well as to test those new compounds I just ordered. And Dave Stout's crystallography lab is trying to get an atomically detailed picture that proves exactly where these compounds bind and how they bind, the detailed binding mode. So we know where they're predicted to bind, but we need actual experimental proof that shows exactly where they bind and how they bind. They've had some preliminary results, some promising results for a few of these hits, but they need to refine the conditions and get a higher resolution or a more detailed picture that proves exactly where these compounds bind. So I'd like to thank the World Community Grid volunteers like you for giving us this huge amount of resources. If you're not yet a volunteer, you can join us at worldcommunitygrid.org. I'm a member of Art Olson's lab. I also need to thank David Goodsell, Stefano Forley, Sargis Dalakian, Ruth Huey, and Mike Peak. The HIV program project team at Scripps led by Art Olson. So Teresa Sample does the crystallographic experiments in Dave Stout's lab. Ying Chuan Lin does the assays in John Elder's lab, the activity assays. Max Chang does the DSF assays in Bruce Torbett's lab. Mike Box does the BSI assays in MG Finn's lab. Subhash Villaparthi in Valerie Fulkin's lab is currently synthesizing those derivatives. And the FightAIDS at Home project was founded in 2001 by Professor Art Olson and Garrett Morris in a collaboration with Scott Kurowski of Entropia. I'd also like to thank the IBM team that controls World Community Grid listed there. And I need to thank the sources of the funding for this project. We got money from the NIH, as well as money from IBM's International Foundation. So some of the money that IBM's Watson won on Jeopardy was donated to the project, and we used those funds to actually purchase these compounds that our collaborators are testing. Thank you very much for your attention, and we'll now turn it over to the Q&A period.
Thank you, Dr. Pearman, for this interesting talk about the FightAIDS at Home project. I hope those listening who have not already done so will also contribute their spare computer power by joining worldcommunitygrid.org. The next part of the lecture is the question-answer session about the FightAIDS at Home project on World Community Grid. Dr. Alex Pearman will be answering your questions. You can ask them using the chat interface at the right side of your window. The first question is, are these compounds still effective in mutations, or do they inhibit mutation?
At this point, we've mainly been testing them against the wild type because we want to find compounds that bind well and change the conformational preferences of both wild type and mutants. Soon, our collaborators will start testing some of these compounds in the new candidates against the mutants. But the whole goal is to find much more potent allosteric inhibitors that could then be used in combination therapies with active site inhibitors. And in other systems like cancer and also for HIV reverse transcriptase, these combinations of active site inhibitors plus allosteric inhibitors has been shown to suppress the evolution of new multi-drug resistant mutants. So the goal is to find things that would help regulate these flaps and close the flaps, which could potentially counteract one of the main mechanisms that the superbugs use to escape some of the protease drugs by having much more flexible flaps and more flap opening behavior. So if we can find these allosteric inhibitors that close the flaps, then that could pay some of the energetic cost and potentially help rescue the potency of the current protease drugs against some of these mutants. But in combination therapies, it does have good potential to suppress the evolution of new mutant superbugs.
How do you know the drugs won't have secondary effects?
That is definitely an issue with all types of drugs developed for any type of purpose, the toxic side effects. So one thing that they do is when the compounds are being developed, when we're getting much more potent inhibitors, hopefully, then those are tested in different types of assays against human cells or against human enzymes that are involved in or known to be involved in toxicity issues. And so that can be used to give an initial indication of which compounds being developed are more likely to be toxic. Ultimately, you don't know until a much later stage of the project, where the compounds are actually tested first in animals and then ultimately in people. And it's only when you start getting large numbers of those results in real organisms that you start to understand whether they're actually toxic or not, and how toxic they are, and whether that toxicity can be decreased by changing certain chemical features of the compounds.
Once you find the compound, how do you synthesize it?
So all of these compounds that I talked about today were all commercially available. So we get the structures of the compounds from the Zinc server run out of UCSF, and we dock them, and then the ones that seem interesting we then purchase right now through a company called eMolecules. That's kind of like the eBay of the chemical compound world that deals with a lot of different vendors. Now for these derivatives, those are being synthesized by people whose entire job is synthetic medicinal chemistry. What they do is they figure out how you can buy different commercially available precursors and combine those together and then modify them in order to make very specific compounds. The synthetic chemistry is one of the strengths of the Scripps Research Institute. It's one of the best places in the world for figuring out how to synthesize things and how to synthesize them more quickly and more cheaply.
Can you briefly explain how the experiments are done virtually using our computer cycles?
Sure. So AutoDock involves two different steps. There's the AutoGrid calculations and then the AutoDock calculations. So AutoGrid is a program that takes the structure of the target, the three-dimensional structure, or a model of the three-dimensional structure of the target, and then it rolls different probe atoms. So it takes all the different types of atoms that are present in all the different ligands, and then it'll go through and it has a probe atom representing each of those different flavors of atoms, and it rolls them all along the entire surface of the target molecule in a grid-based representation, and then it stores the energetics of that interaction, how positive or how favorable or unfavorable that interaction would be with that type of atom. So that's all stored in these maps that then allows makes it much easier and quicker to then look up how well a ligand should score. And the scoring is done during the AutoDock phase, which involves it's actually a Lamarckian genetic algorithm where the compounds, many different copies of the compounds go around and explore that grid-based representation of those affinity maps in order to try to find good locations and orientations and conformations to bind well to the target. And those different poses are scored using the data from those affinity maps. And basically you go through and it evolves and has multiple different generations where the compounds that score the best, the genes, the alleles for the genes that describe its position, orientation, and conformation for the really well-binding compounds are present at a higher frequency in subsequent generations, so that the phenotype updates the genotype, which only occurs in simulations, not in real evolution. But then over thousands of different generations and many hundreds of copies of these of each compound, you're then able to get a decent idea of where the compound binds, how it binds, and how tightly it binds. And then we search through those results to find the cream of the crop, the compounds that dock the best, and then I visually inspect those binding modes, measure the distances and angles involved, and figure out whether it looks like a potential drug-like molecule.
Is there a way to attach toxin to the key molecules to poison the HIV virus?
That type of idea is explored in other therapeutic areas like cancer research, where you have certain molecules that help direct it towards a tumor, and then that's attached to either radioisotopes or to different types of toxins to then kill the infected cell. Since HIV infects cells of the immune system, using that toxin type approach might not be the best way to go. At this point, HIV is treated with about 26 different drugs that target many different classes of compounds. There are entry inhibitors, or CCR5 receptor antagonists, or reverse transcriptase inhibitors, or protease inhibitors, or integrase inhibitors, and maturation inhibitors. But those all target very specific, they're chemical compounds that try to block the activity of very specific viral enzymes, or the CCR5 receptor antagonists block the human receptor. They basically plug the keyhole of the receptor on the human cells that HIV interacts with in order to infect a T cell. So in general, for at least current ways of treating HIV, it all involves these small molecule compounds that either shut down the activity of specific viral enzymes or that block the ability of the virus to interact with the human cell.
Is the key structure unique to AIDS virus only?
The lock and key type idea, that's really just a gross oversimplification of the way that any type of compound binds to any type of protein from anything. It's was the first general model or theory for how something binds to a biological macromolecule.
Do we think there will be a moment when FightAIDS at home finds a cure?
It's definitely possible, and that's what we're spending a lot of our time and resources trying to do, is to try to discover, or at least try to discover, new candidate compounds that the medicinal chemists and synthetic chemists could then modify in order to give us real potential drugs that would then need to go through animal and human studies and clinical trials to see whether it's an actual drug. But we're working on, especially now with this new HIV center called the Hiv for HIV interactions and viral evolution, working on multiple different types of targets from HIV to try to develop new, very potent compounds that make it harder to evolve resistance against, that work against the superbugs and the wild types, so that we could have new combination therapies that could help treat and potentially cure the current HIV patients, as well as to hopefully slow down the spread to other people. So it is definitely possible at some point in the future that we'll have enough good drugs that are designed in ways that make it harder to evolve resistance against them. So we'll have new combination therapies that could ultimately treat it well enough and drive it down to insignificant numbers in the population. That's the overall goal.
Would it be more effective to develop a drug that locks into both receptor sites?
That has been discussed in the program project grant meetings, trying to link up something that would bind to the 1F1 site and the active site, or the 49 site and the active site. The 49 site's a bit too far away, so you'd have to have a very, very long, flexible linker. And when the compounds get too big, it makes it harder for them to actually get inside a cell, the biodistribution or pharmacokinetic properties. So in general, you want to keep drugs less than 1,000 Dalton molecular weight. So linking these together might not work in terms of that linked compound would be too big to really be able to distribute well within a human body. There might be a way to link the 1F1 site to an active site compound. That's something that we'll explore in the future.
How accurate is the model for predicting actual binding?
Autodoc, as well as all the top docking software out there in the scientific community, they tend to have an error in the energetics of predicting how tightly something binds of about 2 to 2.5 kcals per mole. And in general, they're able to dock most compounds to less than a 2 angstrom root mean squared deviation from the known binding mode. But then again, when you're actually doing those studies to determine how accurate the binding mode is and how accurate the potency prediction is, that's all done with current compounds that they know exactly where they bind and how tightly. Docking totally new compounds against something can be a little harder because of the flexibility of the target and induced fit type effects that occur, where the binding of a particular compound can change the shape and flexibility of the site that it targets. So the actual virtual screens get much more complicated. But in general, those are the standard errors for Autodoc and most other docking programs. So it is definitely not proof at all. So we don't know exactly where these compounds bind. We have predictions that tell us where they most likely bind, and that's why our collaborators have to do these experimental assays to get real evidence that indicates how they affect HIV protease, and the crystallographic studies to prove exactly where and how they bind.
Can a key molecule be attached to immune system agents to enhance their autoimmune capability?
That general idea is part of a field called molecular adjuvant therapy, where you try to find compounds that you either fuse to an antigen or include in the same formulation with an antigen to prime the immune system to recognize that and develop a more effective antibody response against it. So that's not something that we're working on. People that work on vaccines focus on those areas where we're searching on trying to discover new things that can be turned into drugs to treat the infection as opposed to the vaccines to prevent it.
Have any of the candidates made it into production medications yet? And if not, how many more years might it take?
So nothing from FightAIDS at home has yet risen to that level of being a real clinical candidate. So we're focusing on an earlier stage where we're trying to discover totally new types of compounds, new classes of potential drugs against different sites. much later in the project, after we found much more potent binding things, and those have been optimized to bind tightly just to the HIV target and not to human cells, and be able to distribute well in the body, then they could hopefully develop into that. In terms of how long this will take, that's really difficult to predict. A lot of science is hit and miss and luck, and you just have to be persistent and keep trying day by day to advance this research and to share it with others so that they can try to build off of it as well. But knowing how long it would take is not something I'll try to guess.
How many different HIV inhibitors are already in use?
So in terms of FDA-approved drugs, there are 26 different FDA-approved drugs. There are 2 entry inhibitors, 1 CCR5 receptor antagonist, 7 nucleoside or nucleotide reverse transcriptase inhibitors, 5 non-nucleoside reverse transcriptase inhibitors. Those NNRTIs actually target an allosteric site on reverse transcriptase, and that's generated some of the proof from other systems that the combinations of allosteric inhibitors and active site inhibitors make it much harder for the virus to evolve drug resistance against that combination. There are over at least 9 different Hiv protease inhibitors that are FDA-approved drugs, but those all target that active site region in the center. So we're trying to find new classes that would bind elsewhere and that would help increase the potency of those currently approved active site inhibitors. There's also one Hiv Integrase inhibitor. Hiv Integrase is a target that we also focus on in some of these FightAIDS at home experiments and in some of our future plans. Oh, actually, there is one. The maturation inhibitor is actually an immune system affecting molecule called alpha interferon. And so alpha interferon actually kind of recruits and activates parts of the innate or primitive immune system, as well as having some effects on the adaptive immune system. So that one, there's only one drug against HIV that's FDA approved that really does focus more on regulating the immune system and increasing its ability to fight HIV, and that's alpha interferon.
We have one more question. How much of the population would need to receive the medication to effectively inhibit AIDS worldwide? Would this be part of eradicating AIDS?
Yeah, I would say that all of the infected people need to be taking the heart treatment, the highly active antiretroviral therapy drug cocktails. They need to be taking it as soon as they know they have HIV, and to take it every day, as many times a day as they're told to take it when they're supposed to take it. And if patients take all the drugs that they need every day when they're supposed to, then that actually also helps decrease the evolution of new mutant superbugs. And recently it was shown the idea of a treatment as prevention, that when patients are taking all the proper anti-AIDS drugs, that their risk of infecting other people decreases by about 96%. Now, it doesn't completely block transmitting HIV to other people, but when infected patients are taking their drugs and the drugs are working against the strains that are present and evolving in that patient, then that does decrease the chances of infecting other people. So when we have better detection efforts and people know they have HIV, and if they're all able to take the proper drug treatments, then that will help us eventually significantly decrease the number of people who have HIV in the world and the rate at which it spreads. There are also totally other types of research that focus just on the ability of preventing new HIV infections in terms of vaccines as well as antiviral microbicides that are showing some potential but aren't quite effective enough yet.
Well, thanks again, Dr. Alex Pearman, and all those attending. Sorry if we didn't get to your question. Please use our forums at worldcommunitygrid.org to post any additional questions. This concludes our webcast.